Firstly, thank you to Nick and all of the conference organization team, and a very special thank you as well to Gordon for being a terrific host and having a wonderful time here in Croatia. So I hadn't fully appreciated just how large the screens were going to be here, so apologies if the next slide looks a bit terrifying. It's a fairly graphic illustration of aging, highlighting clear loss of hair, decreasing eyesight, and an inability to smile, apart from one or two occasions. So some of these are, particularly hair loss is very much genetically driven. However, there are other aspects of the aging process where lifestyle and the environment are more important. And in my group, we're very much interested in both genes and the environment and their interaction to improve the prediction of disease so that we can either prevent or delay the onset of these diseases later in life. To do this, one of the main cohort studies that we focus on is called Generation Scotland. The study began in 2006 when we recruited over 24,000 individuals to a cohort. People were living all across Scotland, and fairly uniquely to the cohort, we have a fairly rich family structure, so up to 4 generations per family. The age range at the start of the study was 18 to 100, and we're currently recruiting new volunteers to the study with over 7,000 individuals having volunteered this calendar year. Crucially for the talk today, Generation Scotland is the largest published DNA methylation resource in the world, with around 20,000 individuals on whom we have Illumina methylation. It's a pretty big multi-omics resource in general, so we have genetic information, epigenetic information. A few weeks ago, we got mass spectrometry proteomics on the 20,000, so that's also the biggest mass spec protein data set in the world. Through collaboration with Gordon, we've generated glycomics on 1,000 individuals, and we also have data linkage to electronic health records. So this is to the GP records, hospital records, prescription data, and it helps us look at the molecular phenotypes with disease outcomes. So covering some of the work that Steve's already discussed in the previous session, just going to skim through some of the first, or what we call first and second generation epigenetic clocks. So these all rely on the striking association between DNA methylation and chronological age. So here we have a single CpG site, so one point at which we can have methylation in the genome, and we see a striking association with chronological age. These are data points for around 4,500 unrelated individuals in our cohort. This slide's a little bit on the dry side, but kind of important in how we actually generate these predictors of epigenetic age. So we start off on the top there with around 800,000 CpGs that we can measure on these arrays. We then use statistical machine learning to ask what is a fairly simple, so a small number of CpGs that can accurately predict our outcome. And here the outcome would be chronological age. So we run this, we get weights for some of the CpGs. Other CpGs we just throw out or we give a 0 weight. And then that formula then gives us a prediction of somebody's epigenetic age. So in Steve's 2013 publication, he showed that the epigenetic age predictor correlated very highly with chronological age in 8,000 samples. Earlier this year, in collaboration with Steve as well, we bumped up the sample size to 24,000. So in epidemiology, really, sample size is king. And as we increase numbers, we get better predictions. So in Steve's original study, the median error was around 4 years. But in our study, when testing an external data set, the median error was 1.7 years. So in lay terms, this means that in the population of 2,000 that we looked at in the test set, for 1,000 of those individuals, we're able to predict their age to within 1.7 years. However, as we get increasingly accurate predictors of chronological age, is this still a biomarker of aging, or is it just a predictor of somebody's calendar age, and perhaps more useful in a forensic context? So what does it mean in the example here if we have a 40-year-old with an epigenetic age of 65? Is this giving kind of cellular aging? Is it an increased risk of certain diseases? And that's really where Steve had the inspiration to explore and develop clocks such as GrimAge. So here, similar to the previous slide, we have lots of CpGs to start with, but instead of predicting chronological age this time, we're predicting the levels of, or sorry, Steve was predicting the levels of plasma proteins. Then in the second stage, he took those predicted plasma protein levels and asked, do combinations of those plasma proteins predict, do combinations of those plasma proteins predict the outcome mortality? And that's how he derived the GrimAge calculator. So we've already seen this, the wheel explaining GrimAge, and when we applied this to some of our data sets, Steve has already covered this as well, that what we saw for the 25% of individuals who had the fastest acceleration or the oldest biological age, around half of them died over 15 years of follow-up. And that was compared to just 10% of the population with the lowest biological aging based on GrimAge. So I mentioned that GrimAge is made up of various different methylation proxies for proteins. Some that I'd like to highlight, as I'll come back to them later, are the one in turquoise on the left there, so GDF15, C-reactive protein, which is, where's that? In the purple on the right-hand side, and then also the smoking predictor on the bottom in red. So with the second generation of clocks, we've seen that they're strongly predictive of overall all-cause mortality, but what about risk of disease? And this is where we took advantage of the data linkage capacity in Generation Scotland, where we have good records on hospital and GP data going back to 1980. So anytime an individual saw their GP, codes were written down, we can extract that information and look at their disease history. So Daniel and the team recently compiled this for over 300 different disease outcomes. These are the most commonly diagnosed diseases from that list of 300. So hypertension being the most commonly diagnosed outcome at a mean age of 60, but with quite a wide standard deviation there. So taking a random set of the 4 disease outcomes, we see here the variability in the age at which an individual was diagnosed. I've also shown here, again, possibly a bit dry, in the red dotted lines, the 2.5 and the 97.5 percentiles. So we did this to kind of exclude individuals beyond those extremes, primarily because some of the diseases in the list are age-related, so kind of a way to kick out outliers, if you like. So we wouldn't want to consider early onset Alzheimer's disease in a study of Alzheimer's disease, necessarily anyway. So we ran a variety of epigenetic clocks against these incident disease outcomes. And the 2 examples shown here are peripheral arterial disease, and on the right, heart attack. And you can see that GrimAge shows a very strong association with time to onset for each of these diseases. And this is over 10 to 15 years of follow-up. And across all 300 disease outcomes, we haven't analyzed it statistically yet, but eyeballing the data, it seems that GrimAge performs extremely well and better than the other epigenetic clocks that we focused on. But what about GrimAge and GlycanAge? So in collaboration with Gordon and Adam Butterworth in Cambridge, we generated glycans on 1,000 individuals on whom we also had methylation data. And it's lovely actually to see the replication of the correlation that Gordon showed earlier. So in the bottom right here, this is a very small correlation between your biological acceleration based on glycan age and GRIM age. So in other words, if you have a biological aging or biological acceleration of around 5 years on GRIM age, that doesn't necessarily mean that you'll have a 5-year acceleration based on glycan age. So in a smaller subset than the 20,000, so just in the 1,000 individuals, some preliminary analyses from Rob in the group was looking at the association of glycan age, GRIM age, and some other clocks with COPD on the left. So we see a very large hazard ratio for GRIM age. But on the right-hand side, we see that glycan age actually outperforms or shows a stronger association with type 2 diabetes than GRIM age, which, given the nature of glycans, one might expect. Interestingly, both GRIM age and glycan age acceleration show similar associations with mortality. But again, important to note that this is a fairly small number of events, just 48 cases of all-cause mortality over the 10-15 years of follow-up. So in addition to building these biological clocks, or these clocks for biological aging, in the lab we've also asked, can we directly train predictors of disease outcomes? So Yi Peng published this study in Nature Aging earlier in the year, where instead of having the methylation predicting age, we're now asking, can we directly predict type 2 diabetes, again through the data linkage from the methylation? So he trained a predictor in Generation Scotland, tested it in Generation Scotland, and also tested it in a German cohort called CORA. And what he saw was that the methylation predictor for diabetes is increasing the predictive performance over and above traditional risk factors. And that replicated in the German study as well. So again, possibly a bit of a dry slide here, but important if there's anybody interested in the methods of the clock generation. So when I mentioned that we have 800,000 CPGs, this can be quite a lot for training models, and ideally, we would want to reduce that number to try and prevent overfitting. And one way we've done this, or Yi Peng has done this, is to first identify CPG sites that are associated with traditional risk factors, so alcohol, smoking, BMI, cholesterol. And then we take that smaller subset of CPGs and feed it into the bigger training models. And we see that this actually outperforms pretty much all other methods that we've looked at for predictor generation. So what can we do to extend and refine possible inputs to GRIM age? In the next few slides, and given that we have the gala dinner tonight, going to dedicate those slides to smoking and alcohol consumption. So here, this is some work by Ola in the group, where we've tried to see if we can improve the methylation predictor for pack years of smoking. And as Steve mentioned earlier, we see a very, very powerful prediction of smoking from methylation data, to the extent that we can discriminate current smokers from never smokers with near perfect accuracy. And again, just by the simple trick of increasing the sample size, so from a few thousand individuals up to about 17,000 individuals, we're able to generate a predictor of smoking that correlates more strongly with pack years of smoking than the GRIM age smoking component, so the correlation jumping up from 0.66 to 0.75. Interestingly, for the smoking methylation, we also have access to some post-mortem brain tissue. This is from 14 individuals from the Lothian Birth Cohort. And what we see across 5 different regions of the brain. And what we see on the left-hand side of the box plots here, in the top left, we have a CPG site where, again, there's pretty clear, so just from one single CPG site, very clear discrimination between never smokers and current smokers. On the bottom, we see not an awful lot happening across the different smoking categories. But then conversely, when we look in some of the brain tissue, albeit with very small samples here, sample size, for the same CPG site in the hippocampus, we see no discrimination of smoking status in the top right-hand panel, but in the bottom we see a pretty clear discrimination between the smoking categories. So the take-home message from this slide might be that the impact of smoking on the blood is different from the impact of smoking on brain regions. For alcohol consumption, again, we've applied the same trick. Just using the large sample size, we've asked how well can we predict an individual's self-reported units per week. And we see that it correlates around 0.42 in 2 external populations. But more interestingly here, I think, is the right-hand side of the slide, where we've looked at the self-reported units of alcohol per week and the methylation proxy for units per week in relation to brain imaging data. So we have here total brain volume, gray matter volume, white matter volume, and the red bars there show the associations with self-reported smoking, where we don't really see an awful lot going on. However, with the methylation-based proxy for smoking, we actually see associations in the direction to indicate that increased alcohol consumption has a negative impact on brain health. So in addition to refining some of the predictors, we can also simply just increase the number that we want to look at. So earlier, or last year, we published a paper in eLife where Danny, a PhD student in the group, generated 109 methylation proxies for different proteins, and she looked at those in relation to disease outcomes. We've also had GDF15 measured in all 20,000 individuals in Generation Scotland, and again, compared to the GDF15 proxy in Steve's Grimage paper, we show that in our independent test sets, we're increasing the predictive power from the correlation of 0.2 to 0.28. Importantly, I think this is also the first study to compare the measured proteins with the methylation proxies for the proteins with disease outcomes, and we see very similar associations. Finally, some ongoing work from Rob has looked at methylation proxies for C-reactive protein. Again, it's exactly kind of the same trick as the previous studies, although here we've been replicating the association across different ancestry populations, and it's nice to see that the methylation score is quite portable across age ranges and different ancestry groups, so including a cohort in Singapore where there's a mix of Indian, Malaysian, and Chinese ancestry individuals. So where does this all lead to? So I think you could maybe picture something like this, where we have, for each of the traits I've measured, whether it's grim age, alcohol consumption, smoking, you could get a score, which puts you in a percentile category relative to others in a population. And that kind of gives you an indication of an individual's general health based on methylation profiles. But I think there's also something a little bit naive in the work that we've done and in relation to disease outcomes, and that is that we look at individual diseases in isolation, when in reality that's not how these things happen. So we know diseases co-occur, and Scott, who was an absolutely fantastic master's student in the team, he's built a platform whereby we can enter all of the GP data, the hospital data going back to 1980, and then we can ask what transitions appear between different disease outcomes. So we can end up in a situation where we have, a wee bit small to see here, but if we have stroke as the outcome, we can ask what diseases happened and what disease transitions do we see in the population prior to that stroke outcome. Or we can flip it around the other way and say, once a stroke has been diagnosed, what other diseases happen further down the line? So it's really refining the prediction of these epigenetic clocks and protein markers. Can I ask, how am I doing for time? is that 30 minutes? It's just say join today and enjoy Netflix. So it's not.
You have 10 more minutes.
10 more minutes. Oh, excellent. Okay, so I will now, with apologies to Gordon, take my feathers off and show some work on protein predictors in UK Biobank. So in Biobank, there's 50,000 individuals in whom O-link proteins have been measured, so around 1,500 proteins. And Danny in the group looked at the associations between, or asked, can we build protein scores or protein clocks for different outcomes? She did this for 23 common or leading causes of morbidity and mortality, adjusting for age and sex, and then in the test set asking what happens when we throw in lifestyle covariates, and then again when we throw in an extended set of kind of biological biomarker predictors as well. And what we see here is that the protein scores, the protein clocks, are actually adding quite a bit on top of the additional biomarkers that we have. So again, it's kind of the same story across whether in Generation Scotland with methylation, UK Biobank with proteins, that if we use omics data, we can improve the prediction of disease outcomes. So in summary, I hope I've convinced you that epigenetic clocks associate with multiple different health outcomes. And I think kind of interestingly as well that we can use some of the associations between methylation and proteins, smoking and alcohol to impute these variables into studies where they've not already been measured. So you can imagine studies where people have collected a blood sample or a saliva sample, but they haven't asked detailed questionnaires. If you derive methylation data from those samples, you can then kind of impute some of these variables into your study. And finally, should we be looking at protein clocks, glycan clocks, or methylation clocks? I'm going to massively cop out and say that more data are required, generic statement. But I think particularly it would be nice to see glycans, proteins, and methylation in a large cohort on the same individuals so that we can really look to see whether there's additive contributions, which my guess is there will be, and for which diseases the different omics platforms give us best information. So finally, just to thank everybody in the team, and in particular, all of the volunteers from Generation Scotland, the Lothian Birth Cohorts, and all of the other studies that we work with. Thank you for listening, and happy to take any questions.